So peace be upon you all, and welcome to a new episode of Seeing God in Biology with Dr. Fuzz Rana. And today we have a fascinating topic, which is cell membranes. And maybe cell membranes is one of those fascinating things that lend themselves to a designer, right, Fuzz?
Yes, I think so.
Yeah. So it's going to be split in two parts. The first one, which is about origins, and the second one would be pondering more on the design itself. So let's see what you've got for us today, Fuzz.
Yeah, sure thing. Well, I think when it comes to the case that people make for the design argument from biochemistry, a lot of times people focus, rightly so, a lot of attention on the structure and the function of proteins, the structure and the function of DNA, things like DNA replication, protein synthesis, the design of the genetic code. And oftentimes people just completely overlook a very important biochemical system, which are cell membranes. In my PhD work and my postdoctoral studies were in cell membrane biochemistry. And so I think cell membranes really are a neglected area for us to make a design argument. And so I hope people that watch this episode and the following episode will really fully appreciate just how remarkable cell membranes are. I think people that do have some familiarity with cell membranes probably don't appreciate just how complex and how sophisticated these systems actually are. And in these two episodes, we're really only going to be scratching the surface in terms of the complexity and the sophistication of these systems, but hopefully it's enough for people to get a feel again for how remarkable these systems are, and why I think that just simply looking at the nature of cell membranes alone is enough to convince us that there probably is a mind that is behind life itself. So anyway, that's a bit about what we're going to try to do here in these two episodes. The first thing to point out is that cell membranes are made up of two major classes of molecules. One class are called lipids, and people may or may not be familiar with that term. But lipids are a structurally diverse set of compounds that has as its primary feature water insolubility. These are compounds that are soluble in what are called nonpolar solvents, and they are insoluble in water. And because of that, it's again a structurally diverse set of materials that fall into this category. Now, even though the term lipid may be unfamiliar to some people, lipids are again very important molecules inside the cell that play a role in membrane formation, which we're going to talk about. But they also serve as a means to store energy for the cell, and they're involved in what are called signaling pathways, where information from the environment or from one region of the cell to another is communicated from biochemical system to biochemical system. Some familiar examples of lipids would be fatty acids. These are the materials that are used to make up soaps. And in the cell, fatty acids are used for energy storage, as well as serve as building block materials to make more complex lipids that play a number of different roles inside the cell. Triglycerides, people oftentimes will visit the doctor and will get a report of their triglyceride levels. These are, again, familiar lipidic materials, and these are used primarily as a means to store energy for the cell.
Lipids, for the audience, lipids are like these fatty, oily things, right? So
yes, exactly. Yeah. They're fats. They're fatty, oily things. Cholesterol is another example of a lipid, and again, we're concerned about cholesterol levels in our blood, but these are very important molecules inside the cell that serve as components for cell membranes, and likewise are involved in signaling. There are certain genes that are turned on and turned off through the use of steroid hormones, which are derivatives of cholesterol. The molecules that we're going to spend a lot of time talking about would be phospholipids. A common name for phospholipids are lecithins. So maybe if you're into health stuff, you might have heard of the term lecithin. These are one of the primary components of cell membranes. And then there are things that are called glycolipids, which are a combination of lipids with sugars that play, again, a number of roles in the cell. Now, another component of cell membranes are proteins. And so we've talked a bit about proteins. We'll revisit proteins in the context of cell membranes here in a minute. So these are the two major groups of molecules that interact to make up cell membranes.
So lipids would be very essential for the cell. Otherwise, if the surface of the cell is made of a soluble material, then the watery medium of the cell would be really a big problem, right? So it will just dissolve.
Yes, exactly. Yes.
Yeah.
And we'll get into. I think we'll get into the. Well, probably not in these two episodes, but in an episode we have in the future about the design of water, we'll talk about why lipids are so ideal for making things like cell membranes. But we'll discuss that here as well in just a minute. But cell membranes, again, serve a number of vital roles. Of course, they form the boundary that segregates the interior of the cell from the exterior environment. So that makes, I think, a lot of sense. But because cell membranes form this boundary, if materials are going to go in and out of the cell, they've got to pass through the cell membrane. And the cell membrane is, generally speaking, impermeable to water and impermeable to water-soluble materials. But it's permeable to materials that are what we would call hydrophobic or nonpolar materials. And so part of the means by which materials get in and out of the cell through the cell membrane is through the use of proteins, which function as transporters. And there's a variety of different types of protein transporters. Cell membranes have proteins on the surface that function as receptors that bind molecules that are found in the environment. And those binding events can then trigger signaling pathways that lets the cell know what's happening in the exterior environment. And then cell membranes also play a critical role in harvesting energy for the cell to use. So cell membranes are vitally important. And just given their central importance, if you're looking to make a design argument, I think it's perfectly reasonable to think that because of their central importance, that these two should be systems that, again, reflect the creator's handiwork.
Yeah.
Oops. Now, this is a cartoon showing a small piece of a cell membrane. So this is a kind of a cutaway. And this, again, is a cartoon, kind of an artistic rendition. But the defining feature of the cell membrane is what is known as the bilayer. And as the name implies, it's essentially consists of two molecular layers that kind of stack on top of each other to form a matrix that is, in effect, two molecules thick. So this is the major component of the cell membrane. And then embedded in the membrane are a number of different proteins, different types of proteins. So the protein composition of cell membranes is incredibly diverse. And this diversity of proteins associated with cell membranes allows the cell membranes to carry out a variety of different functions for the cell. So it's one of the most metabolically active regions in the cell because of, again, the fact that it is serving as a boundary and is communicating what's going on in the environment to the cell, but also because of the transport processes that are taking place. Now the major component of that bilayer are materials called phospholipids. And there's an arrow showing a phospholipid in
Yeah.
that diagram. And so this next diagram gives us a bit of a blow up of a phospholipid. And so phospholipids, if you go to the left-hand side of the diagram, are complex molecules, actually. They consist of a glycerol backbone. So glycerol would be 3 carbons where each carbon has an OH group attached to it. And glycerol is a common material that I think most people are familiar with. And to one of the OH groups on glycerol is a phosphate group that's bound. And to that phosphate group is bound an alcohol. So there's a phosphodiester there. That is considered to be the head group region. The glycerol to is considered to be the interfacial region of the molecule. And then to 2 other OH groups are attached through an ester linkage, are attached these hydrocarbon chains. And so so the left gives you essentially a sort of a sense of the molecular structure. The central image is giving you a space-filling model of what the molecule looks like. And then on the far right is a cartoon representation of a phospholipid. So for example, when we go back to this diagram and we see the bilayer with the little balls and the little tails, that's what's being represented in that diagram. And by the way, biochemists refer to these kind of diagrams as supramolecular diagrams, where it gives you a sense for how the molecules are interacting with each other, where the diagram is depicting just the overall molecular shapes. So you get a more a larger a macro perspective on the how the molecules are interacting. Now, this is again a depiction of a bilayer structure where we're not looking at any proteins in association with it. And again, we can see in the right-hand corner that the representation of a phospholipid. Now, because the phospholipid has a head group that is made up of a phosphate and an alcohol attached to it, it is water soluble. And the tails are water insoluble. The hydrocarbon tails are water insoluble. So when you add phospholipids to water and they're above what's called the critical micelle concentration, they will spontaneously assemble into this bilayer structure, where the interior of the bilayer is hydrophobic, where the hydrocarbon tails do not want to interact with water, so they're able to be sequestered from the water, whereas the head groups are water soluble and want to interact with the water. So it creates this thermodynamically stable structure that spontaneously forms because of the nature of the phospholipid structure. So, and we'll talk more about that in the next episode when we talk about the design, because the structure of the phospholipid molecule, every feature of its structure is precisely the way it needs to be for bilayers to form. And there's not really alternative options that you have. Now, this is just to give some context, this is a diagram showing on the left a fatty acid molecule. So a fatty acid would be one of the molecules that combines to form the phospholipid. So the fatty acid would be shown here as that portion, that hydrocarbon tail, that long chain that is bound to the OH group. This is through, again, an ester linkage where the carboxylic acid reacts with the alcohol to form what's called an ester linkage. And so this is just the fatty acid by itself. But because of the molecular geometry, and we'll talk more about this in the next episode, because of the molecular geometry, fatty acids are also what are called amphiphilic, where part of the molecule is water soluble, part is water insoluble. But instead of forming bilayers, when fatty acids go above the critical micelle concentration, they actually will assemble into these spherical structures called micelles. And this has to do with, again, the nature of the packing of the molecules. But that gives you, again, a little bit of a sense for these kinds of molecules. Now,
so you will not find a cell membrane that is actually a fatty acid micelle. You need the bilayer to have the big wrapper around the cell. Otherwise, the fatty acid micelle will be just this small sphere with a microscopic gap in the middle, which because I've heard in many discussions that I've gone through, people will say, what's the big deal about cell membranes? Just spill some fat into some water, they will form those micelles, which is like a proto cell membrane or something, and then maybe it will trap some other material inside, and with a strike of luck, maybe the material that gets trapped inside would be just the right material, and then evolution or something would take over, and this kind of argument. So can we respond to that? This will actually come later.
No, this will be in this episode. That beautifully sets the stage.
Okay, good.
But the teacher in me, the biochemist in me, just wants to spend a little bit more time talking about, because I think again, I as a membrane in lipid biochemist, I interact with biochemists who are not that familiar with cell membranes. They're only superficially familiar with cell membranes, and they tend to think of these as just being these haphazard structures that spontaneously form when phospholipids are present. And the complexity and the sophistication of these systems is far greater than I think most people appreciate. So I'm hoping to, in the next few minutes, just really begin to convey just how remarkable now these systems are. Now, when it comes to cell membranes, the phospholipids actually form a highly diverse population of molecular species. Even though we see, again, phospholipids forming the bilayer, there's a lot of molecular diversity to phospholipid structures. So, for example, the hydrocarbon chains that form the hydrophobic interior of membranes, the fatty acids that are bound to the alcohols of glycerol, can vary in length. They can actually vary in degree of unsaturation, where if you introduce a double bond into the chain, it's going to cause the chain to kink. If you actually introduce multiple double bonds, you actually can get the chain to actually curve the more double bonds that you introduce. So you actually have variability in terms of the chain length, as well as the whether the chains are linear or kinked or even curved to some degree. And then in addition to that, this is not a very good diagram, I apologize, but also the alcohol that's bound to the phosphate group can differ. So, for example, if you have no alcohol bound, that's called phosphatidic acid. If you have a glycerol, that's called phosphatidylglycerol. If you have ethanol amine, that's phosphatidylethanol-amine. You can have choline. You can have inositol, which is a polyalcohol. And so the point is that you can vary the head group structure by the identity of the alcohol bound. And so for example, the type of alcohol that you have bound is going to determine the charge of the head group. So for example, glycerol is neutral, but because the phosphate group is a -3 charge, of course, one of those charges is taken up because you have the phosphoester linkage. But if you have glycerol, which is also forming an ester linkage with the phosphate, it'll have a -1 charge. You can have serine, the amino acid serine bound, and that actually gives you a -2 charge to the phospholipid. You can have ethanolamine and choline, which gives you a positive charge, which makes the phospholipid overall neutral, but it's still a zwitterionic species because of the negative and the positive charge. So the point is that you can, by having these different alcohols, you have different charges and different sizes of the head group, and different chemistries for the head groups. So there's quite a bit of variability when you start looking at the combinations of head groups and fatty acid chain lengths and degrees of unsaturation. So when you look at the number of molecular species in the bilayer in a cell membrane, it's an enormous number. You can start beginning to think through all the different kinds of permutations and combinations that you would have here. There's an enormous amount of chemical diversity that we see in cell membranes. And the temptation is to think of that chemical diversity as being haphazard, as being, it's just a very sloppy, haphazard system, which is oftentimes how people think of cell membranes. But in fact, as we're going to see, there's a high degree of order to cell membranes in spite of this high degree of molecular diversity. Now, let's see here. This is
the challenge. The challenge now is that if any phospholipid can actually do a bilayer autonomously or spontaneously, then how come if there is a zoo of them that we actually have the right ones all the time? This would be the question. If the right ones are there all the time, it lends itself to design. If it's just a random zoo, then okay, maybe something else. Yeah.
Right. Exactly. Okay. So now, one thing that is really important to appreciate is that there's actually what's called an asymmetry to the distribution of the phospholipids in the bilayer, where the composition of one leaflet is different than the composition of other leaflets. And so it's not just again a random mixture of phospholipids, but their compositional distribution is precisely controlled by the cell. By the way, this is so. For example, in this diagram, we see in the upper leaflet or the upper monolayer certain phospholipids that occur at higher frequencies than in the lower leaflet or the inner monolayer. Sometimes they're called the outer monolayer, the inner monolayer, the outer leaflet, the inner leaflet, where the inner leaflet is interacting with the cytoplasm, the outer leaflet is with the extracellular space. And there's this diagram shows some other lipids that we aren't going to talk about, sphingomyelin and glycolipids. But what we can see is that, again, there is an asymmetry in the compositional distribution, where certain phospholipids with particular head groups are concentrated in the outer leaflet, and then the inner leaflet has phospholipids with different head group structures. And this is actually vitally important to the function or the functional diversity of the cell membrane. Now, also, there is the phospholipids in the bilayer are not static, but they actually can diffuse throughout the bilayer. So this is a highly dynamic system that we're dealing with. And there are different kinds of movement that are possible. So, for example, you can have what are called rotational movements, where the phospholipids can rotate. That, right? They can laterally diffuse through the monolayer that they're in, and then they can also undergo what's called a transverse diffusion or a flip-flop motion, where they can flop from one leaflet to the other. Now the rotation and the lateral diffusion occur very rapidly. Those are very rapid movements for phospholipids. The flip-flop motion is actually a very slow motion. In fact, it's highly restricted, because what happens is that highly charged head group would have to pull itself into the hydrophobic interior, which is thermodynamically not preferred, in order to go from one side of the membrane to the other. So that flip-flop motion is largely restricted, which is how the bilayer can then maintain its phospholipid asymmetry because of that restriction in that movement. And so here's another diagram showing again kind of the idea of the flip-flop motion, where the flip-flop motion is restricted. It's a very, very rare event when you get a phospholipid to spontaneously undergo flip-flop motion. But there are proteins in the cell membrane that actively will facilitate that flip-flop motion. And these are called flipases and flopases. Very, very cleverly named.
Interesting. Flipases and flopases. Okay, that's
So, but the idea is that these proteins are actually playing a critical role in establishing the phospholipid asymmetry and maintaining it. So that asymmetry is essentially is designed and is regulated by the cell's machinery. It's not, again, haphazard. It's highly intentional, where these enzymes are actively maintaining, again, that phospholipid asymmetry.
So those machines, they pass around in the cell membrane, flipping actually the phospholipids from one side to the other to maintain a certain structure or a certain differential between both leaflets.
Yes, and there are enzymes that and that flip-flop motion is actually requires energy to be used to order to move the phospholipids. So the flipases and the flopases are actually energy utilizing enzymes, and there's some enzymes that will move the phospholipids from the outer leaflet to the inner leaflet, others that'll move it from the inner leaflet to the outer leaflet. So the direction of the movement for a particular enzyme is actually controlled. So there's a whole population of these flipases and flopases. And then there's these enzymes called scramblases, which actually will move phospholipids in both directions rather indiscriminately. So you've got a pretty sophisticated collection of enzymes that are working collaboratively to maintain this phospholipid asymmetry, and then to modify it if it needs to be modified in response to something happening in the cell or in the extracellular environment. Another point is that bilayers have a physical state as well. And so bilayers can either be, sometimes they're called liquid crystals because there's a three-dimensional long-range order to the bilayer, but that, again, because of the ability of the bilayer, the phospholipids, to diffuse laterally, it actually is like a liquid as well. So it's like a liquid crystal, but the bilayer can have regions that are more gel-like and regions that are more liquid-like, and so that you can vary the physical state of the cell membrane, and this is all due to the way in which the hydrocarbon chains of the phospholipids pack. So if you have longer hydrocarbon chains that are saturated, they tend to pack together very tightly, and that tends to create kind of a much more viscous region of the membrane. If you have shorter chain lengths, or if there is unsaturation, you don't get as good packing, and that creates a more fluid region in the cell membrane. And again, the factors that influence fluidity would be the chain length of the fatty acid, the degree of unsaturation, and then cholesterol is a major component of cell membranes, and it functions like a plasticizer. So if the membrane is fluid, by adding cholesterol, it'll make it more gel-like, and if it's gel-like, adding cholesterol will make it more fluid-like. So cholesterol is playing, again, a very important role. So this is showing how cholesterol fits in there. And again, you can see why it would again, because
Mhm.
it disrupts the packing, it makes it
Mhm.
more fluid. And
Yeah.
because it, again, if the packing isn't great, it kind of acts like a bit of a glue to create more a gel-like structure. So by varying the cholesterol level and the type of sterol, there's, in addition to cholesterol, there's other sterols that are in cell membranes. By adjusting the composition or the type, you can actually control the fluidity or the physical state of the cell membrane.
Yeah. So essentially, if it's more fluid, does that make it more permeable? That's what it does.
It can.
Yeah.
The fluidity, typically, the more fluid it is, the more permeable that region is. But also, the proteins that are embedded in the membrane through their catalytic cycle have to be able to breathe. They have to be able to
Ah,
undergo these movements. And
move.
so,
Yeah.
in regions that are not very fluid, the enzymatic activity
Or
for
difficult.
is more difficult. In more
Mm.
fluid regions, the activity can accelerate. So you can regulate enzyme activity by regulating the fluidity of the membrane.
Wow. They're very, very smart. Very smart design.
Now, when it comes to the proteins that are connected to cell membranes, there are, broadly speaking, two types. One are called peripheral proteins. These are typically attached to the surface of the bilayer. And then there are proteins that are called integral. Some integral proteins span the entire bilayer. Some of them are only embedded into one of the leaflets, one of the
Mm.
monolayers.
Mm.
Uh, and again, membrane proteins are what mediate all the different functional activities of cell membranes. And this is a diagram showing, again, some of the functional roles that proteins are playing. Some of them are transporters, where they are actively pumping materials across the membrane against the concentration gradient. So if you
Ah.
want to accumulate certain nutrients in the interior of the cell, then you need to actively transport those materials from the exterior environment into the interior cell against the gradient. So this would be called active transport.
So, for
Sometimes,
example, the medium has a low concentration of a certain salt, but you need this salt, so it keeps pumping it in,
right,
regardless that it is more concentrated inside than outside anyway. So, for example,
right,
if you leave it for something like osmosis, for example, or diffusion, it will level out, but those transporters keep pushing them in
right.
to capture more of this nutrient or whatever it is.
Yes, that's
Yeah.
right. And then
Yeah.
now some transporters are called passive transporters. They
Mm.
basically are pores that are going to allow the flow of materials across the membrane based on the concentration. So
Mm.
that movement follows the concentration gradient. Um, some proteins are receptors that will bind against small molecules, triggering other biochemical pathways that function as signaling pathways. Some are enzymes that will convert molecules on the surface of the membrane from one molecule into another.
Mm.
And then there are some proteins that are called anchor proteins that actually anchor the cell membrane, for example, to the cell cytoskeleton. And then
Mm.
some of them are actually anchoring the membrane to extracellular molecules. So, for
Ah.
example, in multicellular organisms, you
Mm.
have this extracellular matrix, and that membrane, the cell is anchored to that matrix through these anchor proteins. So
Mm.
these proteins are doing a lot of different activities for the cell.
Wow.
And the model for the cell membrane that was proposed in the 1970s is called the fluid mosaic model. So this was proposed in 1972. And the view of the membrane here then is you've got this bilayer matrix of phospholipids that's kind of like a sea where the proteins are just floating around. The proteins
Mm.
are diffusing, through the membrane, through the bilayer, either as integral proteins or even those proteins that are attached to the the surface, the peripheral proteins, they still would migrate around because they would have this attachment to the phospholipids at the surface of the membrane. Now, that idea, however, is being replaced or has been replaced by what we might call the new fluid mosaic model, where this particular view sees the membrane as actually a highly organized system. Even though, again, the complexity is bewildering in terms of the number of different proteins, the number of different phospholipid species, there's an organization that people are now recognizing to the membrane. So, for example, we talked about phospholipid asymmetry. Well, there's also protein asymmetry, where certain leaflets will have higher concentrations of certain proteins, the other leaflet will have a different concentration of proteins, or the proteins may be form local concentrations where certain proteins are found in certain regions, localized regions of the cell membrane, and other proteins are found in other localized regions. There are these things called annular lipids, which are lipids that form a close association with membrane proteins, forming essentially a layer around them, and that layer serves as a lubricant to give that protein the freedom to move in order to carry out its catalytic activity. And then there's now this recognition that there are actually domains in the bilayers where certain regions differ from other regions in terms of fluidity, phospholipid composition, and protein composition. And so, in fact, there's this idea that's still a bit controversial that there may actually be what are called lipid rafts, where you actually have regions of the membrane that are basically highly, where the phospholipids are highly, where you have a precise phospholipid composition, a precise protein composition to that region, where that region actually carries out a specific biochemical task for the cell, and that the physical properties of that region keep it kind of intact as like a raft floating on a sea of phospholipids. And so here's a kind of a computer generated depiction of a cell membrane surface. So this is just showing you one of the leaflets. And you can see that the different colors, where the purple represents, the kind of the bulk bilayer, where you have these regions that have localized concentrations of certain phospholipids that differ in fluidity from the bulk bilayer, so that because of that they remain more or less intact, and they actually then float around or move around as a unit within the cell membrane. And so this is another depiction of kind of a lipid raft, where you have particular proteins that are associated. And in fact, sometimes lipid rafts, if they're of the same type, can actually float around, interact with each other. They might break apart and then diffuse away from each other. So you're looking at a highly ordered system, but it's also, again, a highly dynamic system as well. So this is just kind of scratching the surface. We could spend a long time talking about the details, but I think hopefully this is enough to help appreciate just how complex cell membranes are, right? And how sophisticated that complexity is. It's not a haphazard complexity. There's a long range order and a short range order to the structure of cell membranes that is intimately linked to the functional properties of different regions of the cell membranes.
Wow. So even this cell membrane is like a world on its own. It has modules, it has regulation, it has machines, it has ways to control those machines to work or not work, or be activated or not. And It
Right.
even this matter of those anchoring guys is just amazing. So the cell, in a way, is aware that it is part of a bigger group of cells, and there's this skeletal structure that it needs to hook to.
Yes.
So the design of the cell is not only inner looking, but it's also appreciating that it's part of a bigger whole.
Yes.
In a mechanical sense, this is like it's a beautiful idea that the cell, it's like it knows that it needs to hook to something because it's part of a bigger organism or something.
Yeah.
And this hook is there as part of its function. So
Yeah.
it's again one of those chicken and egg kind of thing.
Exactly.
Because I have always, and I have gone into some of those discussions with some, unguided evolution kind of thinkers, and I always challenge them when it comes to the transition between unicellular and multicellular organisms. And my question is, why on earth would a cell that lives independently find any path to become a part of something else where it loses its own independent life?
Right.
And since the thermodynamics would tend to make things simpler rather than more complex, it doesn't make sense, even in terms of physics.
Yeah.
And then to find this in the design of the membrane itself is just it's just amazing. It's
Yeah.
So,
Well, and we touched, as we talked about these different aspects of cell membrane structure. I think I could already see the wheels turning in your mind, Ahmed, about thinking through the design implications. And so we'll pick that up in the next episode.
Yeah.
Where we'll revisit some of these features, and then we can speculate and elaborate on why we see this as evidence for design. But I thought what would be good is to talk a little bit about how did cell membranes originate, because this is part of the origin of life problem. And again, when I've looked at the work that goes on by origin of life researchers, there's a lot of attention paid to how does metabolism emerge, how do information-rich molecules emerge, with, as you said earlier, where people are just of the opinion that once you start getting fatty acids and maybe phospholipids or something that's a intermediate between a fatty acid and a phospholipid, it's just going to assemble spontaneously into a micelle, into a bilayer, and voila, you've got a compartment. So people have largely neglected studying cell membranes. There's been a few origin of life researchers who are really membrane biochemists first, who recognize that this is really short-sighted, that the origin of cell membranes in and of itself is a real problem, is a real challenge, and that needs to be addressed. And so what we'll do is we'll talk a little bit about some of those challenges, because from my perspective, the origin of cell membranes is just as challenging a problem as how do you explain the origin of an RNA molecule. It's just as challenging of a problem. And when it comes to, a chemical evolutionary, view for the origin of cell membranes, there are a number of steps that have to be generated. The first is there's got to be some mechanism by which you would produce or deliver, the materials that would make up cell membranes. And by the way, most origin of life researchers think that the very first cell membranes were primitive, being made up of primarily fatty acids, and that it was later on that phospholipid-based membranes would have emerged. So they see the contemporary membranes that make up, cells today as being something that is really beyond the reach of chemical evolution, and that the very first membranes, again, would be primitive, made up of fatty acids. You have to have some mechanism by which those materials self-assemble. into bilayer structures. And this is not an easy step, by the way, even though it's often portrayed as a very easy step. It's not by any means. You've got to have a means by which you can encapsulate, let's say, self-replicating molecules or metabolic systems. You've got to have a way to move materials in and out of the cell. You have to have a means by which the cell membrane can function in energy acquisition. This is one of the really critical roles of cell membranes. Cell membranes primarily function as an energy generating system for the cell because the bilayer itself could be thought of as a capacitor, right? So if you have a concentration differential of charged materials across the membrane, let's say protons are at a higher concentration across the membrane than they are on, or higher on the outside of the membrane than on the inside of the membrane. That charge differential is going to be maintained because of the impermeability of the bilayer to protons. And so it's functioning like a capacitor where you now have a battery, right? And if you can have a means by which to collapse that charge separation, you're going to create a current flow, which is essentially what happens when you complete the circuit, when you have a battery, right? And so like enzymes like the F1F0 ATPase use that proton gradient across the inner membrane of mitochondria. That motor component actually creates the conduit for protons to flow.
Wow.
And by those protons flowing through that motor, that's generating essentially generating electrical energy that can activate the rotation of the rotor.
Wow.
And so in other words, when it comes to energy generation, that is a very important aspect of the cell membrane that has to be accounted for in an origin of life scenario. And then, of course, you've got to have the means by which cell membranes can grow and divide. So these are, and then you have to explain the transition from primitive membranes to contemporary membranes. Now, in terms of where do fatty materials come from? Well, there's a reaction called a Fischer-Troph reaction that was discovered by two German chemists, as the name implies, and maybe in the 1800s, where you can take carbon monoxide and hydrogen, and at very high temperatures in the presence of metal catalysts, you can form hydrocarbon chains. And if you add carbonate to the mixture, you can actually get fatty acids. So origin of life researchers have speculated that maybe this might be a reaction pathway that could generate fatty materials on the early Earth. The problem is that this is requires gaseous molecules, and it requires basically metal catalysts. So it's hard to envision where this kind of chemical environment would exist on the early Earth. And now there are some people that have shown that you can use oxalic acid as a carbon source and hydrogen and carry out this kind of reaction in an aqueous environment. And so they speculate that maybe these kind of materials could be formed at hydrothermal vents. But the problem is that this reaction is contaminated by hydrogen sulfide. And so hydrogen sulfide would have also been produced at these vents. So in terms of the biosynthesis of these materials, it's not really that evident where long chain hydrocarbon materials would necessarily come from. Now, there are some people that think they may have been delivered to the Earth extraterrestrially. So for example, if you look at the organic extracts of the Murchison meteorite, you actually do see fatty materials. There are lipidic materials that come out of it. But But the question is, are you going to be delivering enough on the early Earth for it to make a difference? Some people have shown that if you take these simulated comets, and you irradiate them with UV radiation, that you can generate, again, fatty materials that, again, have speculated or have fueled speculation that maybe it was an extraterrestrial delivery. So right now, it's still an outstanding question as to where these materials come from in and of itself. But let's just say, for the sake of argument, you grant that there is a source of these materials. The next question is, how can these materials form, bilayer structures? Because remember, we talked about the fact that fatty acids form micelles. They don't form bilayer structures.
Yeah.
And this is actually really important, because a micelle might be able to, might be able to sequester hydrophobic materials, but there's no aqueous internal space. And so you're not going to get aqueous-based biochemistry in a micelle.
Yeah, right? Because the region inside will be pretty small. It will just capture a few things inside, but not really a medium, right?
Right. And it's only going to be create an environment for nonpolar materials. So the nucleotides that you would need to build RNA, for example, would not be soluble in the micelle interior. They're going to prefer the exterior environment. So you really need a vesicle where you've got a wall where there's an aqueous interior. Now, this is a electron micrograph of extracts from the Murchison meteorite, that the lipidic extracts that under certain conditions in the lab will actually form these vesicle-like structures. And so this has fueled speculation that maybe fatty acids on the early Earth could actually form vesicles and form kind of like a primitive protocell. And so the idea here is that you've got at the top basically the air-water interface. This is hypothetically on the early Earth. And that you would get these lipidic materials, these fatty acids, either formed at hydrothermal vents, or they would be delivered to the Earth through some kind of meteorite or cometary delivery. And because of the hydrophobic nature of these materials, they're going to accumulate at the air-water interface, right? This is they form a monolayer at the air-water interface. This is well-known phase behavior for these lipids. And then that some of those would then dissolve from that air-water interface into the bulk aqueous phase. And at a high enough concentration, they would form micelles. And if you can get the just right set of conditions, those micelles would form into bilayer vesicles that could then serve as, again, the first protocells that could encapsulate materials. So this is, the model for how this would work from a chemical evolution.
Those are still fatty acids, right? They're not phospholipids.
Yes, fatty acids. Yes.
Yeah. And will the vesicle in this case be stable, so it will just keep staying there as a vesicle, for enough time that needs to be quite a considerable time for anything significant to happen inside it?
That is a very perceptive question, and the short answer is no. And I'll explain why that is in a minute. But but this is how people envision the formation of the very first primitive membranes. Again, these are fatty acids. And part of the problem is that phospholipids, it's a real struggle to get phospholipids to form under any conceived environment on the early Earth, because first of all, phosphates will react, or not will react, but will form complexes with calcium and magnesium that are insoluble. So calcium phosphate and magnesium phosphate are insoluble. So any calcium and any phosphate on the early Earth are going to interact to form these insoluble salts. Fatty acids also will form insoluble complexes with calcium and magnesium as well. And so the early Earth environment it's such that you're not going to be able to even have these starting materials available.
Because of the contamination with other minerals around them. Yeah.
Exactly. And let's say you found an environment where that was pure water, where there were no, ionic materials. You have to, in order for phospholipids to form, you have to drive them. They only will form under severe dehydrating conditions. You almost have to drive every molecule of water away from that system in order to get phospholipids to form. And so most origin life researchers say, look, they concede we're never going to get phospholipids initially on the early Earth. The only way so we have to go to fatty acids as really the very first types of membranes, where later on you might be able to introduce phospholipids through some unknown mechanism, to go towards a contemporary cell membranes. Now, this, your question anticipates this next slide, because the fatty acid vesicles only form under a very precise set of conditions. First of all, they only will form once the fatty acid is of 8 carbon lengths or greater. So C7, C6, C5 will not form vesicles. Now, the problem is that when you look at the distribution of molecular components, like in the Murchison meteorite or through cometary delivery, the distribution is you got a very high concentration of, 1, 2, 3, 4, and 5, carbon fatty acids, and the concentration decays exponentially as you go to longer and longer chain lengths. So you have to have at least a C8 or C9 fatty acid to get vesicles. The concentration in the Murchison is going to be extremely low.
Okay.
Now, in addition to that, fatty acids have a relatively high critical micelle concentration. So in other words, and it's at a millimolar level. So in other words, unless you have fatty acid concentrations in the bulk phase above a millimolar level, they're only going to remain dissolved as individual molecular species. They're not going to aggregate into micelles or into vesicles. So that's another problem. Now, in comparison, phospholipids have a critical micelle concentration, I believe, in the nanomolar range. So these things are strongly bilayer forming, whereas fatty acids are not in and of themselves. And then you've got to have, again, I'm not sure what I meant there by chemical composition. You can actually get greater stability of the vesicles, and they form with a little bit greater ease if you incorporate fatty alcohols into the mix. The problem is you have to be a just right fatty alcohol fatty acid ratios. The pH is absolutely critical. You have to have a pH that is at the pKa value, so that you have roughly a 50-50 mixture of the ionized fatty acids and the unionized fatty acids. And
essentially you need a lab.
Yeah, exactly. And there can't be any salt in the system. It has to be basically distilled water, or nearly distilled water. So yes, fatty acids will form vesicles, but it's under an extremely precise, exacting set of conditions. And so the idea that these things are going to form vesicles is really a pipe dream. You can, again, as you said, in the lab with highly skilled biochemists, you can coax the system to do this, but it doesn't do it very easily on its own. Now, for encapsulation, people have a pretty good mechanism where if you have, let's say, RNA molecules that are dissolved in solution, you have, let's say for the sake of argument, vesicles. If you dehydrate that system and then rehydrate it and dehydrate it and rehydrate it, over time you will create encapsulated materials. The problem is, because the formation of fatty acid vesicles is so finicky, to undergo dehydration and rehydration, you're never going to wind up with vesicles. If you somehow had a solution of vesicles, once you dehydrated and rehydrated it, you're never going to get the precise set of conditions again that you would need to form the vesicles. So while there is in principle, a mechanism that would work, it isn't going to be a very valid mechanism, whatsoever.
So if you even turn out to get the vesicles, to capture things inside the vesicles means that you will lose the vesicles themselves through the hydration, rehydration, dehydration process. So it beats itself essentially. I would either have the vesicle or the vesicle will capture something and I lose the vesicles.
Yes.
A chicken and an egg again. Okay.
Yeah. Yes. Exactly. Now, also, and again, we even when you're working with phospholipids, actually are much more readily will encapsulate materials in fatty acid vesicles. You can make phospholipid vesicles, but even then, you have to have a strict ratio of fatty acid. Of phospholipid to the material you're trying to encapsulate. The salinity and the pH can actually be very important as well when you're trying to do encapsulation experiments with phospholipids. So even though phospholipids are more amenable to this, they still, again, require an exacting set of conditions.
Apart from the fact that just to get phospholipids in the first place, doesn't seem to be something that will happen in a prebiotic setup by anyone.
Right. Yes, exactly. Yeah. But even if you somehow got them and you were able to form vesicles, you still have the problem of, again, the encapsulation is still a fine-tuned process that's much more likely to happen in a lab than on the early Earth. Now, permeability is something very important because once you've got the protocell, you've got to have ways for materials to go in and out of.
Yeah, you need all the transporters and all the portals and the receptors and the whole bunch of things which wouldn't be there, right?
Right. Now, this is where fatty acids actually have some advantage because fatty acid vesicles seem to be far more permeable than phospholipid vesicles. Phospholipid vesicles, as a rule, are impermeable to a lot of materials. And the only way you can adjust the permeability is through fine-tuning the molecular composition of the phospholipid species. If you tweak it, you can get a set of conditions where permeability is possible. So, but fatty acids, again, are much more amenable to permeable, are much more permeable. But now another problem, though, is having to do with energy acquisition, because again, remember we talked about the fact that you've got to maintain a gradient across the membrane. And this is where fatty acids are problematic, because in a fatty acid bilayer, the flip-flop motion is very easy to take place. All right, whereas it's not so much in a phospholipid bilayer. And the reason why is because the fatty acid, the carboxylic acid, can become protonated, and as a protonated carboxylic acid, it's going to be readily soluble through the bilayer interior. And so it readily undergoes a flip-flop motion, but that flip-flop motion is coupled to the acid-base reaction that the carboxylic acid head group takes place. So imagine you've got a proton gradient across a fatty acid bilayer. The fatty acids that are exposed to the higher concentration of protons are going to react with the protons to form a protonated carboxylic acid.
And then flip inside
and then deliver that proton. So the fatty acids will dissipate any gradient that you have established. So there's no way to create a gradient across fatty acid vesicles.
While in the case of the phospholipids, they will keep the protons outside until the capacitor effect takes place, and then you can have currents and use currents to do different things. Yeah.
Yes, exactly. So now another problem is, how do you transition from let's say you somehow have these protocells made up of fatty acid bilayers. How do you then transition to the contemporary membranes that we see in biochemistry today? And we already talked about the fact that to get phospholipids to form under the conditions of the early Earth are so challenging that, again, almost to a T, origin of life researchers will agree that we have no understanding as to how to do this, right? So this is not even disputed that phospholipids would be a struggle to form. Now, another problem is that if we look, let's say we somehow have a mechanism for phospholipids to form. If we look at bacteria, bacteria have primarily two types of phospholipids: phosphatidylethanolamines, which are abbreviated as PEs, and phosphatidylglycerols, which are abbreviated as PGs. You don't see phosphatidylserine, phosphatidylcholine in bacterial systems. Those are primarily found in eukaryotic cell membranes, not bacterial membranes. But what this is telling us is that the very first contemporary phospholipid bilayers would have to be made up of primarily PEs and PGs. Now, this is a problem because phosphatidylethanolamines have a tendency to form what are called non-bilayer phases. Now, and we're gonna get into this into the next episode, but some phospholipids readily form bilayer structures. Other phospholipids form what are called non-bilayer phases, and phosphatidylethanolamines actually form what are called inverted micelles, where you have the
tails are looking outside.
Right, and so what it is, think of a column that's surrounded by the charged head group, and then you have the hydrocarbon tails pointed away, and then that tube stacks on another tube, so you still have the hydrocarbon chains are sequestered away from the water. It's just that the water phase is now in these tubes of the inverted micelle, right?
So the micelles captures the water inside them rather than protect the tails inside them. So the tails are now outside and the water gets trapped inside. Wow.
Now, by itself, a single tube isn't going to be thermodynamically stable in water, but when you start stacking them together, which is what's being depicted here, now you suddenly have this massive, lipid phase. But the problem is that when you add phosphatidylglycerols to phosphatidylethanolamine, it actually makes the formation of the non-bilayer phase occur more readily. The reason why these bacterial membranes are bilayers has to do as much with the protein composition as it actually does with the lipid phase that you see there. So the fact of the matter is that if somehow you could form phospholipids, again, what we know about bacteria and their lipid composition suggests that the very first bilayers must be made of PEs and PGs, but that's a problem because of these non-bilayer phases. Now, this is something that also is really I think is very interesting. When I was a graduate student, the question that I had probably within a month or two of being in graduate school, because I studied cell membranes, was how do cell membranes actually form? Because the cell membranes are made of what is called a unilamellar phase. It's a single bilayer. Usually when you dissolve phospholipids or you disperse phospholipids into water, you don't dissolve them, but you disperse them, they will actually form bilayers, but the bilayers are not these unilamellar phases. Instead, they look like this. They're stacks of bilayers.
Oh.
Or that are sheets, or they're this is like a cutaway, or they look like onions. right? Where there's bilayer, 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 but it's like a vesicle, right?
Ah, interesting.
And in the lab, you can get these single bilayer vesicles to form. These are called liposomes, but it only will form when you use a certain type of phospholipid, and you've got to basically sonicate the system to form these vesicles that then within a few days collapse back into these multilamellar phases. And so my question as a graduate student was, how on earth do cell membranes form as unilamellar phases when that's not what you typically see phospholipids doing?
Yeah.
And the clue to this came from a scientist by the name of Norman Gersfeld, who actually did a series of studies that basically showed that unilamellar phases are critical phases, meaning that they will form only for a particular phospholipid. They will only form at a particular temperature. So if you have a phospholipid that you disperse into water, it'll form into these multilamellar sheets and vesicles. But if you heat the system, when you hit a just right temperature, you suddenly get a unilamellar phase. And he then showed that the temperature for that unilamellar phase will vary from phospholipid to phospholipid.
Wow.
So it's a characteristic feature of pure phospholipid species.
So once it is fixed into a unilayer for one type, it means the other types will not like it. So
right. Now and then if you have a mixed bilayer system where you have different phospholipid species, you can get that to form again a unilamellar phase, but that composition will have a precise temperature, and you vary the composition, the temperature varies. So in other words, to maintain a unilamellar phase, which is what a cell membrane is, depends upon a precise fine-tuning of the phospholipid composition. And if you deviate that ever so slightly from that value, that unilamellar phase will collapse into a multilamellar phase.
Yeah.
And so Norman Gersfeld did these studies where he showed that the lipid composition of bacteria actually changes as the growth temperature changes. So if you have E. coli and you grow E. coli, let's say at 30 degrees Celsius or 37 degrees Celsius, the membrane composition actually is different for E. coli at those two growth temperatures, where the bacteria actively adjusts its membrane composition to maintain a unilamellar phase in light of the growth temperature.
Wow. That's mind-blowing. So the bacterium would adjust the composition of its membrane so that the membrane remains a membrane in that specific temperature. Essentially, it has a computer in its pocket, and it knows the different compositions for the different temperatures. The temperature changes, it will change the composition just like that.
Yes.
Wow.
Yes.
So it has the scientist notes with it.
That's right. Another experiment that Gersfeld did was with sea urchin embryos. And sea urchins live in the ocean. They're cold-blooded creatures. And depending upon the temperature that the sea urchin embryos were in, again, they would adjust their membrane composition to maintain a unilamellar phase. And what he showed is that when you actually heat red blood cells, they will undergo basically hemolysis, where they'll break apart. And when they break apart, oftentimes that unilamellar phase will end up collapsing into a multilamellar phase upon red blood cell hemolysis driven by temperature. Because what you're doing is you're forcing the red blood cell out of a temperature regime above 37 degrees Celsius that is not compatible with its unilamellar phase, and the red blood cell will burst apart, and again, and you form these multilamellar sheets. Wow. So anyway,
can I ask a question here? Because there is something that I'm expecting. I'm not sure if it's true or not. But would it be true that the abundance of proteins in the cell membrane will make the sensitivity of the unilayer to break up or collapse into a multi-layer, less sensitive? I mean, it is not just by varying the composition in terms of the phospholipids, but the availability of the proteins themselves can be a stabilizing factor or a regulating factor, at least one way or the other. Is that something that somebody has researched or not?
Yeah, I think that is probably true. That it might broaden the temperature range that the cell can tolerate, and remain in the unilamellar phase. But in these 3 studies that he did with, authentic, biological membranes, like with bacteria, the sea urchin, and red blood cells, they are, populated with the proteins that would naturally occur.
Yeah.
And so they still lose that the unilamellar phase
under enough heat stress, under enough temperature variation. Yeah.
Yes. Yeah.
That's the very fact that the cell would change the composition to adapt to the temperature to a certain limit, of course, obviously,
right,
is on its own mind blowing. Is there any understanding of how or where the cell keeps the recipe of the right composition given the right temperature? Is this uncovered yet?
If it has been, I'm not aware of it. So
Okay.
and part of what I see in outside of people that work on cell membranes as biochemists is that most people, again, are not even aware of the degree of fine tuning that you need to have in terms of the molecular composition of cell membranes in order for the cell membranes to function. I don't think people fully appreciate that. But that fine tuning, means that to go from, again, a fatty acid bilayer to a phospholipid bilayer is no easy task, right? You have to pay attention to, the phase structure, making sure that you're going to form bilayer phases. And even then, you have to have a precise lipid composition to form these unilamellar bilayers. And so the transition from primitive to contemporary, cell membranes is really very difficult to envision how that could ever happen under the conditions of the early Earth because of the fine tuning that's involved.
Amazing.
So in fact, a few years ago, I wrote an article with a chemist by the name of Jackie Thomas that was published in Origins of Life and Evolution of the Biospheres, where all this stuff we just talked about, we actually presented in a scientific journal article that was accepted by Origin of Life researchers as being, it went through a peer review process and was accepted for publication. Where we basically, the title of the paper is The Influence of Environmental Conditions, Lipid Composition, and Phase Behavior on the Origin of Cell Membranes, where we're making the point that all of these precise conditions that are needed in order for these chemical evolutionary scenarios to work for the origin of life represent a very real obstacle to chemical evolution. And until there's an understanding of how to overcome this, you're never going to have a valid model for chemical evolution. So anyway, so what I presented to you has actually been published in the peer review literature. So it's not just something a couple of ID friendly people are cobbling together, as a way to complain about evolution. This is actually something that has been peer reviewed. So,
very, very, very nice that actually this went through. Good that no one had future sight of using it in this episode.
Yeah, that's right. Well, it's an interesting story. I had the idea to write this article. And actually worked with Jackie to put it together. And I ended up meeting an origin of life researcher named David Deemer, who did quite a bit of work on the origin of cell membranes from a chemical evolutionary standpoint. And so I told him about that I was writing this article. Would he be interested in taking a look at it? And he agreed. And so when I shared the article with him, and he, to my surprise, he came back and he said, if you do this and this to the article, I think it actually would strengthen it. And and he goes, and I'm thinking that this is where you want to submit the article. I know the editor. I'm going to write him a letter and letting him know that you're going to submit the article. So he actually, and I was critical of a lot of his work in this paper, but he actually advocated for me, and the editor-in-chief of the journal knew that I was a creationist and was fine publishing the work. And in fact, after it went out for peer review, came back and said, here's some suggestions. I really hope that you modify the abstract because we would love to publish this. And so that was one of those scenarios where they respected the critique, and they felt like the critique was actually on solid scientific grounds, and so they were willing to actually be generous and publish it. So there are open-minded people on who hold to an evolutionary perspective, who are open-minded and will acknowledge legitimate criticisms.
Yeah, hope there is more of them with time. God willing.
Yes.
Yeah. So that was quite a ride about the origins, and I guess next part we'll be talking more about the design, right?
Yes.
So for everybody who has seen and enjoyed this as much as I have, please do give us your comments and even throw some questions at us. And hopefully, God willing, next time we'll be looking at more of the wonders of cell membranes, which, like any system, so any system in engineering, you cannot call it a system without, having a certain boundary to it, right? And then the boundary would allow or not allow exchange with the environment is something else. So the system can be like, closed or open, or other definitions, depending if you're talking about its physics or thermodynamics. But at the end of the day, without a boundary, you cannot know where something starts and ends. And what cell membranes are telling us is that these things are not just like wrappers, like random wrappers. They are very well designed to make the cell remain a system with certain boundaries, even with the conditions change to the maximum of the knowledge of the cell, to even vary the composition of how it is wrapped to remain wrapped, and to use the environment to its advantage to collect the materials that it wants, to create gradients that are defiant to the typical entropy or thermodynamics that should govern it, to the lowest energy state. So it's amazing how these things are there. And if this doesn't show that the existence of life had an intentional designer and creator behind it, what else would? I think it's a very obvious case, isn't it?
Yeah, I agree. The organization, the fine tuning that you see, to me, really points to the idea that this is something that is, requires a mind, that requires appropriate forethought and understanding of the chemistry and the physics in order for it to come into existence.
So with this as a closing note, maybe next time we'll see what else did this mind put into the design of this amazing thing called the cell membrane. So with that, I bid you all goodbye, and looking forward to see you all again next week as we pursue this amazing and intriguing ride. Thank you, Fuzz, very much, and looking forward to see you again.
Yep. Thanks, Ahmed.
Thank you.